let's take a look at osmosis, and let's take a look at osmosis and remember that it's the movement of water. Okay, it's a movement of water across a membrane. Now, why do we have to actually look at osmosis as something different from diffusion? Because of that semi-permeable membrane. And often, all too often, osmosis actually acts different from diffusion. In fact, does the exact opposite of diffusion. If we look at osmosis, and let's take a look at the example that we did in the lab, or at least that we tried to do in the lab. Let's take a look at an egg. The egg looks simple. It has a nice shell. It has a nice oval shape. It's actually the largest eggs are the largest cells in every vertebrate's body. And here's a chicken egg. It's normally, by the way, they're not white. They're this brown color. Let's take a little closer look at it and take a look at the anatomy of this egg. The chicken cell anatomy is complicated, much more complicated than we would normally think. You have all different kinds of layers to the chicken egg. The blastoderm actually being the actual cell, the chicken that's going to grow up is right here at this thing called a blastoderm. The yolk has a lot of nutrients in there for that blastoderm, the nutrients that it's going to require to grow, the amino acids the chick is going to need is here in the albumin, and we talked about the amino acids earlier last week. But it's this annoying little part here. The shell is the part that we have to get rid of so that we can access this natural layer that we call a membrane. This membrane is semi-permeable, and in fact is very much like the membrane of the cell itself. Okay, so we got rid of the egg shell by leaving it in vinegar. Any kind of acid would do. The acid eats away at the calcium carbonate. That's the outside of the shell. That calcium carbonate produces carbon dioxide when it comes into contact with an acid, and the shell goes away, and the gas actually. And what you're left with is a membrane, and that membrane is obviously this all around the outside of the egg. You still have the other parts of the inside of the egg yolk, and I'm not even going to try to pronounce this word, but I'll give it a shot. The shalazae. In any case, these internal anatomy parts of the egg, are irrelevant for our experiment, because what is important is that you have a membrane that's semi-permeable. Remember, we talked about semi-permeable, and what that means is that, again, only certain things are going to get in and certain things are going to get out. In this case, only water. In all our experiments, only water will get in and out of this cell. So since water is the only thing getting in and out, that gives us an advantage. That gives us something that we can study and use this model, this egg, to look at what we're really interested in, which is osmosis, or the movement of water across that membrane. So this is what happens to the egg when you put it in 3 different solutions. And this solution here is what happens when you put it in a hypertonic solution. Here's what you have when you put it in an isotonic solution. So there's basically no change. And this is again what happens when you put it in a hypertonic solution. We'll go over what all the hyper and hypo and iso means. And this egg, which seems a little bigger, is what happens when you put it in a hypotonic solution. Hypo, iso, and hyper. Obviously, hyper means a lot, iso means equal, and hypo means low or less. Less what? Solute concentration. It's that solute concentration is what's going to be important here. So it's that How much solute is dissolved in that water? That's going to be so important. We're trying to find out what happened to that poor little egg here. What's the difference here where this mass is, this is almost round, where this one was just left to be on its own as just a regular normal egg. This is a closer look what the two eggs look like when they were outside of, when they were no longer in an isotonic solution. Remember the egg shape. This is almost round. This is a round shape where it's almost completely lost its egg shape that would be more of an oval, right? This is much more round than it would normally be. Of course, this is almost completely void of all water. It lost almost all its water. That's intense. And that loss of almost all the water inside the egg has flattened it out. The only thing you can see is the internal anatomy, the egg yolk, the chaiste or whatever. You can see it here, kind of little wiggly line over here, little wiggly line over here in the yolk outline here. But all the water's gone. It's intense when this lab actually works out well. So this movement of water, this osmosis, is something you have to understand, because this cell is not the only one that osmosis occurs in. Osmosis occurs in every cell of your body. So let's look at osmosis. Let's look at osmosis in three different conditions. The isotonic condition, that's the condition where the concentration on the inside, side 1, is the same as the concentration as on the outside. Because they're the same, any movement of water into the cell is equal to the amount of movement of water outside the cell. So osmosis, there is no change. So your blood cells, these are your blood cells here, a picture of your red blood cells. They're normal. They stay normal. They stay looking like discs. Notice they still have that indentation here that you see here in red. You see it here as well, and you'll see it here and here. So the red blood cells stay like small discs or berets, if you will. In the hypotonic solution, now you have a lot of salt concentration, or concentration of dissolved solutes here are high. They're high. And because the solute concentration is high inside and low outside, normally the high to low, the solutes would move out, but because the membrane is, again, semi-permeable, all right, that word keeps coming up over and over again. Because you have a semi-permeable membrane, you get water is the only thing that can move. How are you going to equilibrate it? The only way to equilibrate that concentration or get the concentration to be equal on both outside and inside is to have water come into the cell, diluting the inside of the cell, making it more like the outside of the cell. So water moves from the outside to the inside. Notice the direction of these arrows. And of course, what's that going to do to the cell? Well, that's going to swell it up. Now, you see here in Figure 1, you see that the isotonic solution, they're still discs. But in Figure 2, because it's hypotonic, in other words, tonic, by the way, is talking about the solution that it's dissolved in. So it's iso because the inside is equal to the outside. It's hypo because the outside is less than the inside. So anytime you see tonic, you're talking about the liquid that the cell is suspended in. So here we have an hypotonic solution because the outside, the liquid that it's suspended in, it has less concentration than the inside of the cell. So because that inside has a higher solute concentration, the water flows across a semi-permeable membrane, that only allows H2O to pass by. The water goes in and swells up the cell. Now, if the difference is big enough, these cells will actually burst, by the way. It's very possible that in a hypotonic solution, that cell could burst. In a hypertonic solution, the exact opposite occurs. Here we have the outside is much more concentrated than the inside in its solutes. So the only way to, again, because it's a semi-permeable membrane, Your your water molecules are the only thing that can pass through the membrane. And because the water molecules are the only things that can pass through the membrane, you have the water leaving or going from the inside to out. So water's leaving because the solutes can't come in, because normally they would go from high to low solute concentration. They can't in this case, so your water has to come out, and they shrivel up. And here you see the cells, the blood cells, all shriveling up in a hypertonic solution. Remember, the word tonic means the solution, in the liquid in which it's suspended in, and hyper means elevated concentration of solutes, right? All right, so let's go on and take a closer, even deeper look into this. So as you learned in elementary school, there's two kinds of cells, or in general kinds of cells. There's many more, but you'll learn more about that later. The key here is that there's at least two different kinds of cells, and that's the two kinds that everybody always likes to talk about are the plant cell and the animal cell. The animal cell has just the membrane. There's no cell wall. The plant cell has a cell wall. Well, that cell wall actually makes a big difference. The animal has in a hypotonic solution, as I said earlier, if there's enough difference, if you're in, for instance, distilled water, what's going to happen is that water is going to start rushing in, keep going in until the cell bursts, and of course the cell dies. We call that lysed. And you can remember, lysed means breaking up, so the cell breaks up. So here in a plant cell, though, that's a good thing to have a lot of water on the outside, and so the plant cell actually likes it very much, and you get a lot of turgid, something called turgid. So there's a pressure that doesn't break up like the animal cell, because it has that cell wall all the way around it, and that cell wall stops the pressure that's building up inside the cell from bursting the cell. It keeps the cell nice and stiff. Now, in an isotonic solution, that's when you have some salt on the outside, some salt on the inside. They're actually pretty equal. In an egg cell and a human cell, the cell, we would call that normal. Whatever normal shape that cell would have, that's isotonic, is perfect for the animal cell. In plant cells, the plants become flaccid. They become a little bit weak. Their cells, there's water going in and out, but you don't have that pressure that you would have pushing up against the cell wall, keeping it nice and stiff. In a hypertonic solution with an animal cell, the animal cell shrivels up, and make it enough of a hypertonic solution. In other words, make it high enough concentration of solutes on the outside, and you're going to get the cell to shrivel up and finally die. In a plant cell, this is horrible because the plant cannot. All the water's leaving the cell. The inside of the cell shrivels up, and this cell just plasmalizes, or just ends up just shriveling up and dying. So plant cells really like it, a hypotonic condition. Animal cells like it, an isotonic. Nobody likes hypertonic. I thought I'd give you one more look at this distilled versus sugar water, just because I thought this picture actually had a better picture of the distilled water, because notice how round the egg is, very round. Remember, distilled water means that there's no solutes in the water, so this is the hypo, right? And that means that there's not a lot of solutes in the water, where this is the hyper, where you have a lot of concentration of sugar outside, not a lot inside. So here you see the egg shriveling up, and here you see the egg almost perfectly round, swelling up to an extreme amount. So the two concepts of osmosis and diffusion really they're very similar, and it's important that you understand the difference. In diffusion, you're dealing with molecules that spread all over a large area. You put a sugar cube into water, it's going to spread throughout the whole area. You put salt on one side of a permeable membrane, not semi-permeable, but permeable membrane. then this all will go across that membrane, and they'll equilibrize, they'll become an equal concentration on both sides. In osmosis, you're talking about molecules that go through a semi-permeable membrane, but the only molecule that goes through is water. That makes it important because of what we looked at earlier. The water is moving, so it has to equilibrate so they could swell up the cell or shrink the cell, so that becomes an issue. When you're talking about what are the similarities between the two, in both cases you have molecules moving. In both cases you have molecules moving from one side to the other on a membrane or across a solution, but the key is that in both cases you're looking for an equilibrium, an equal movement of molecules from one side to the other, so that there's no net change. There's that word net again that we talked about earlier in the year. It's going to come up again. Understand it. No total change. There's going to be some change. There's going to be molecules moving right, molecules moving left. But when you look at the total change, it's a zero net gain or loss.